Hello AP Bio students who are remote. I am making this video to go over the things that we have covered so far in class on Chapter 9 on both Tuesday and Thursday. Some of the first work that we did was with the cell cycle introduction, and that's going to be the first couple slides I talk about. And what I had people do for homework for Thursday's class, which was today, was to make sure that they could draw out with either different colors or different yarn the different aspects of mitosis in terms of genetic information, microtubules, nuclear envelope, plasma membrane, centrosomes, actin protein belt, using these sources and answering these questions about each stage. We are going to go over briefly the notes that we took for that class. And then today we talked about cancer and cell cycle regulation in terms of the checkpoints, cyclins, the cyclin pathway, and also what it means for proper cell division. I did send out a study guide in an email earlier this week. The test is going to be on next Thursday. There'll be a 45-minute question section on multiple choice from chapters 2 to 9, 45 minutes on FRQs from chapters 2 through 9, and then an ecology section that everyone's going to take afterwards. And each chapter is organized based off of some I statements that I think you should be able to answer to show mastery with different aspects of the chapter, whether it's multiple choice or FRQ. Right now, it looks like we are going to have a study session Monday after school, probably at 3 o'clock, to help us prepare for the exam. So let me know if you can make that. So thinking about the notes that we took, these two slides are going to be useful for the first note sheet, and then these notes are for the second sheet that I have for you. So let's get going. An interesting thing to note about cell division is that if we look at some cells, such as E. coli, which are a bacteria, they duplicate very easily because they have a circular chromosome. They don't have a lot of them, and their life cycle is quite short. So if I started with one of these E. coli cells and they divided every 20 minutes, so 1 became 2 and 2 became 4 and 4 became 8, pretty soon I would have what's known as exponential growth. And it would only take about 36 hours for that one E. coli dividing continuously every 20 minutes to become a 1-foot layer deep of bacteria, because of the rate of mitotic division. And pretty soon, 39 hours afterwards, we would have an 8-foot layer deep of bacteria, which is pretty outstanding. So the cell cycle is how bacteria and cells in humans divide and grow. And there's 4 basic aspects of the cell cycle. The first is G1. Cells can be in what's known as G0, where they're never going to divide again, but many cells exist in this G1 phase, which is normal cellular life. They were derived from mitosis and cell division, but they're doing things like they're doing cellular respiration, they're having enzymatic reactions, they're producing proteins, they're basically living life. And at some point, however, in a cell's life, it's going to receive information from tissues or organs or other signals that they need to divide to make 2 new copies of themselves. This could be because you're injured, you have a cut, you need to grow because you're in puberty. It could be that the life cycle of the cell has been reached and it needs to be replaced, or in many bacteria, it could be due to asexual reproduction. And as a result, S phase occurs, where we duplicate all of the chromosomes in the nucleus, and we also duplicate centrioles, which are going to be useful for dividing the cell. And then we get to G2, and G2 is where the cell double checks and prepares for division. It makes sure that there's no mutations in the synthesized duplicated DNA, and then we undergo mitosis. Different parts of your body undergo this process at different rates. Your hair follicle cells are growing at a rate of 0.35 millimeters per day. At points, your teeth stop growing after 12 years. Your brain at parts replaces itself every 20 to 30 years, and some parts it's not. Your heart stops growing after 10 years. Your bones are replaced every 15 years. Your liver, depending on your diet, and things like consumption of alcohol, is replaced every 300 to 500 years, or days, not years. So this cycle occurs all of the time at different rates in cells. So 3 things that you should feel really comfortable with when doing the drawings of mitosis. The first are chromosomes, which are just a physical vessel with hundreds of genes that can be expressed to make proteins. Humans have 23 pairs. In one pair, we have a chromosome from dad and mom, biologically speaking, and they make up what we call homologous chromosomes. They have the same types of genes, maybe different versions of that gene, but they code for the same information. When we duplicate one of those chromosomes and we have 2 copies of one chromosome, we call those sister chromatids. The second thing to be very aware of with mitosis is the centrosomes, which are sort of the organizing complex of mitosis. They produce microtubules, which are protein protrusions that can bind to the chromosomes, and they often dictate the shape of the cell and the chromosome movement. And then finally, the microtubules themselves are polymerized protein strands that are part of the cytoskeleton that attach to chromosomes during mitosis and then help bring them towards the 2 poles of the cell by depolymerizing the microtubules. So we can see different aspects of mitosis occurring in different cells at different parts based on their life cycle. So then we get into the sort of regulation of the cell cycle. And the thing that we first need to know, there are 3 checkpoints for the cell. G2 exists right before mitosis, and it's going to make sure that we have all the duplicated DNA correctly produced. Mitosis has a checkpoint, the M phase, which makes sure that all of the sister chromatids are lined up on the metaphase plate to make sure that 2 daughter cells receive identical information. There's a G1 checkpoint to make sure that cells actually are getting the right environmental conditions to divide before they duplicate their DNA. Regardless of a cell's type, age, or lifespan, all of these checkpoints exist in all human cells. And this might make sense because we all are evolved from bacterial cells, which also undergo the same steps to make sure that they should and can divide. But also we all, in terms of all trillions of cells in our body, came from one fertilized egg that was a single cell that underwent mitotic division as soon as the sperm and egg that created you was fertilized. Now, cyclin is a type of signal molecule, a protein that helps regulate the cell cycle. And different types of cyclins are going to be found in higher or lower amounts throughout the cell cycle. So we have cyclin C, E, A, and B all occurring at different parts of the cell cycle. Now, cyclin C is going to be the first one to start accumulating, and its job is to start promoting the growth of the cell to exit G0 or to enter G1 and start getting ready for division. This is going to start causing the duplication of organelles and to start getting genetic material prepared for division. Cyclin C is going to stop being accumulated as soon as S phase has occurred, so its job is really to get the cell out of G1 and getting the cells into the synthesis phase. Now similarly, cyclin C accumulates until the cell actually begins to develop and copy over all of the DNA in the nucleus to make duplicated sister chromatids. As soon as DNA replication begins, that cyclin E is going to be degraded, which is why those levels drop off precipitously. And their job is to be signal messengers to get enzymes activated that duplicate DNA. Now, cyclin A is going to start accumulating during S phase. It's going to come up quite heavily throughout the S phase, and it accumulates into G2. And it is used to stimulate the duplication of more organelles, to duplicate centrioles, and to start getting that duplicated DNA condensed and organized into a place where we can start prophase. And it's going to be turned off when the cell has duplicated those centrioles, and the DNA is starting to be condensed. And then finally, we have cyclin B, which accumulates all the way back in S phase, but its job is to make sure that the metaphase plate is matched during mitosis and that we have evenly lined up sister chromatids on the metaphase plate. So as soon as we have all of that information and all of those sister chromatids, they're going to degrade. We call all of these processes positive feedback loops because they accumulate more and more and more until a certain response is met. It's sort of like flooding a bathtub. The water is going to continuously flow into the bathtub until it flows over the side, and then maybe you turn off the bath water. So we aren't going to have a degradation or a drop-off in these cyclins until a certain action or threshold is met. Now, in terms of cyclin activity, cyclins are unique proteins because they have a shape and function based off of their R groups, which is determined by their amino acid sequence. And they are going to form a ligand with the CDK or cyclin-dependent kinases, which are receptor proteins. Different cyclins have different CDKs because each cyclin, A, B, E, and C, have a unique shape and based off their amino acid interactions. Once they do form that ligand, they form that association, a phosphate donating protein called a kinase, this is probably kinase A, is going to donate a phosphate from ATP onto that protein. And that phosphate group is a very negatively charged molecule which interacts with all of the R groups in our protein, which sort of flips on and activates the protein. It turns it on to do work. That cyclin D or that cyclin CDK activated receptor is then going to bring the phosphate, tag it to a target protein that is going to then cause more responses to occur, such as a cascading event, which we call signal transduction. So really, the cyclin pathway for all of our different secondary messengers throughout the cell cycle are regulated by receptor proteins, which become activated with ATP because the phosphate functional group is very negative, so it's going to interact with R groups that are positively or negatively charged, which will cause the target proteins themselves to become activated. So let's take a look at an example of one. This is the metaphase checkpoint or the M checkpoint during the cell signal. A couple of things to note about what we can see right here. When we don't have all sister chromatids attached to the kinetochore microtubules, there are going to be some enzymes on the kinetochore region that cause a catalyst reaction or a chemical reaction to produce this complex called MCC. So remember, enzymes are proteins, which means they have an on-off function based off their shape. And if there's no kinetochore microtubule bound to those proteins, the enzyme is activated, so it produces an endergonic reaction by building up MCC. MCC is a molecule that can allosterically regulate this enzyme called APC. And APC is an enzyme that prohibits the breakdown of cyclin B. And it also prevents the enzyme separase from becoming activated. So if we have an allosteric regulation of APC, an enzyme, which means its active site can't bind to either of these proteins, forming a ligand, that means that cyclin B is continuously going to accumulate and the cell is not going to progress into anaphase. And that's because cyclin B, like all of the cyclins, exhibit a positive feedback loop. So we are going to have more and more cyclin B accumulating until we achieve a system where APC is turned off. So once we do have that system where APC can be turned on, it's going to be where the cyclin B is going to be degraded. And what causes that cyclin B to be degraded? It's when all of the sister chromatids are attached and the enzymes on the kinetochore change their functional shape because of that bondage to a microtubule, so they're deactivated. That means that MCC is no longer created and doesn't accumulate. It can't regulate or allosterically turn off APC. So APC is going to be turned on, it's going to be released, it's going to then do two specific enzymatic functions. It's going to bind to the CDK cyclin B complex and break it apart. So cyclin B is going to now start breaking apart and deaccumulating. But it's also going to cause separase to be released from this protein called securin, which regulates allosterically it turns it off. Remember, enzymes can be turned off by having inhibitor proteins bind to it. And that separase, when it is activated, can then go and split up proteins at the centromere region. These cohesin rings, which sort of act like rubber bands around asparagus strands in the grocery store, are broken apart by the separase enzyme, which allows us to then have anaphase occur. So in a nutshell, we can look at how the cell cycle is regulated by enzymatic activity and cell signaling, how receptor proteins and signal pathways cause proteins to be activated, how the accumulation of different signal molecules cause different aspects of the cell cycle to occur, and why those checkpoints are important. And this is all going back to the idea that we need cells to divide in predictable manners that help the body have specialized tissues. I hope this has been helpful. I will see you guys soon.